My name is Jörg Vogel. I'm based at the University of Würzburg, where I run the RNA and Infection Research program at the Helmholtz Institute for RNA-based Infection Research. I'm generally interested in the roles of RNA and how it can, in microbes and in infected host cells, and how they can be exploited for therapeutic treatment of infectious diseases.
We're obviously recording live from the FEMS Conference on Microbiology. So how are you finding that so far?
I think this has been a fantastic conference. I think it's been a fantastic idea to do this in Belgrade. I think it puts places like this on the map. And I've been enjoying to see different faces and seeing different topics on the agenda. So I think there's a much stronger focus on Eastern European countries, which is good because that's a part of Europe that we need to explore better.
A few moments ago you gave your plenary lecture. Can you tell us a little bit about what you talked in that session?
In my lecture I talked about a new research direction in my lab, which are so-called programmable RNA antibiotics, where we use antisense molecules, short snippets of antisense RNA or variants of RNA to target gene expression in bacteria, with either the goal to kill these bacteria or to reprogram gene expression in members of the microbiota.
And I think obviously antibiotics is a very, very hot topic at the moment related to antimicrobial resistance. So how would that affect or maybe improve the way we deal with such infections?
So I think these programmable RNA antibiotics are great because they can both kill the bacteria, but they can also knock down resistance and can resensitize bacteria to pathogens that are drug resistant to existing antibiotics. So I think you can take a two-pronged approach, killing the bacteria through this antisense, by this antisense approach, but at the same time making them vulnerable to the available antibiotics again.
So where do you see this research going in the future?
I think there's a lot we need to understand. I think we're following two directions. One is to try to apply this approach to as many microbes as possible, and I've already got a very positive response, people getting interested in using this against bacillus or other species, species than Fusobacterium nucleatum, the organism I talked about. On the other hand, I think there's a lot of basic research to be done, so we don't really understand what happens when these antisense snippets enter a bacterial cell. And only if we understand how they enter and what happens afterwards, we can prove their efficacy.
And last but not least, what is your favorite microbe?
My favorite microbe at the moment, after having worked on Salmonella for almost 20 years, has now become Fusobacterium nucleatum, a commensal in the human mouth, which sadly also colonizes colorectal cancer cells very well. And so if you swallow Fusobacterium nucleatum or gets in your bloodstream, which happens every day, and enters the colon, it will actually prevent chemotherapy, and it is also associated with a poor prognosis of these patients.
Great, thank you so much for your time and good luck in all your research.
Thank you very much, Eleni.